Hello and welcome back to my channel. In previous lecture we have discussed about DNA extraction, and in this video we will discuss RNA extraction. So without any further ado, let's get started. First we will see what is RNA. As we know that RNA is basically a single stranded molecule that contains ribose sugar. That's why it's known as ribonucleic acid. We also know that it has 4 nitrogenous bases: adenine, guanine, cytosine, and instead of thiamine, in case of RNA we have uracil. And we also know that we have 3 major types of RNA. Messenger RNA, basically that encodes proteins. Second, we have transfer RNA that act as adapter between messenger RNA or amino acids or transfer the message. Third, we have ribosomal RNA that forms the ribosome, small ribosomal subunit or large ribosomal subunit. So here we have a short intro about what is RNA. As we have already mentioned, in this video I will discuss about RNA extraction. So let's see what is RNA extraction. RNA extraction is basically the process of purification of RNA from different samples. These samples may also be blood, tissues, hair, bacteria, viruses, bacterial cultures, etc. So now we will see what are the different components that we require for RNA extraction. So here we have different components that we require for RNA extraction. And in this video we will discuss by triazole method. So the component that we are using for RNA extraction is triazole. Why we are called triazole? Because it includes detergent, that is phenol, that will degrade the protein. After that we have denatured, that is guanidine thiocyanate, that helps in nucleic acid binding or DNA or RNA binding. Third, we have reducing agent, that is beta mercaptoethanol, that will disrupt the disulfide bond between the proteins. Next we have alcohol. We can use ethanol or isopropanol. Next we have wash buffers, that is 70% ethanol, and the elution buffer is RNase free buffer or nucleus free water. So here is the components that we require for RNA extraction. Before going to procedure, I would like to mention here, in previous lecture we have discussed about for any extraction or nucleic acid extraction we have 4 major steps. What are these major steps? These are, first we have the first step that we require for nucleic acid extraction is cell lysis, and for cell lysis we use the lysis buffer. Second step we have precipitation. For precipitation we need alcohol, either ethanol or isopropanol. Then for washing we use wash buffers, and for elution we use the elution buffer. So here we have the 4 major steps that is required for any extraction, either the nucleic acid, DNA or RNA extraction. Now let's move to the procedure that is required for RNA extraction. So let's discuss the procedure of RNA extraction, and in this video we will discuss RNA extraction from triazole method. Why it is called triazole? Because it includes, tri means 3 components like phenol for protein degradation, guanidine thiocyanate for nucleic acid binding, either DNA or RNA, And in the last we have beta mercaptoethanol for the disulfide bond breaking between the proteins. First we will add 1 ml triazole, 200 microliter sample, and this procedure may be vary from lab to lab or from sample to sample. So first we will add 1 ml triazole, 200 microliter sample, then mix well up and down by pipetting. Second, 200 microliter chloroform. Then we will add the chloroform. Then after adding the chloroform, 3 layers are formed. The first layer, aqueous layer, in which the RNA is present. Second, interphase, and the third is inorganic phase. Then after that, incubate on ice for about 15 minutes. Then after incubation, centrifuge. Then transfer the upper aqueous phase to a fresh tube or a spin column tube, because the upper aqueous phase contains the RNA, that's why it is separated in a fresh tube. After that, isopropanol is added for precipitation. Then after that, incubate. After incubation, transfer RNA into a spin column tube and centrifuge at 8000 rpm for 2 minutes. Then change the collection tube and add 70% ethanol and then centrifuge for about 8000 rpm for 2 minutes. Then after that, precipitation, washing 70% ethanol, and then after for elution we will add RNase free water or nucleus free water. After elution, RNA is stored at -20°C. So it's a short review about RNA extraction, and I have already mentioned this is just a review. For further details you can check your recommended books. And I would like to mention here, these protocols or a procedure may be vary from lab to lab or from sample to sample. Thank you and take care.